Hey everyone, this is Dave Inman, the pharmacy instructor. This will be the first lecture of the class. We're doing the pharmacology for pharmacy technicians. We have the third edition of the book. This first lecture, I'm just going to cover the first chapter of the book, and we'll go ahead and post that online for you guys. So if you kind of just walk with me through it, on page 2 we have it starts fundamentals of pharmacology. Pharmacology, by definition, is the study of drugs and their interactions with living systems, including chemical, physical properties, toxicology, and therapeutics. Knowledge of pharmacology is essential to accurate, effective, and efficient performance of the responsibilities of a pharmacy technician. Technicians educated in pharmacology have the skills to properly identify the drug from a patient's profile when refills are requested and the patient does not remember the drug name. Less time is spent searching for drugs when the pharmacy technicians have good brand and generic name recognition. Pharmacy technicians possessing substantial knowledge of the pharmacology can be able to reduce dispensing errors associated with look-alike and sound-alike drugs, incorrect drug strength, incorrect dosage form, and improper dosing schedule. So we'll stop right there and just kind of reiterate again in the course of a daily, your daily practice as a pharmacy technician, regardless of the format really, it is going to be expected. And this happens all day, every day on a regular basis. A customer will call us and say, I'm not sure what I need, but I need my blood pressure pill, or I need my fluid pill, or I need my pill for my heart, my sugar pills. So it is critical for us, for you guys as pharmacy technicians. Part of your role is to know, when they say that, to have some sort of familiarity with at least the most basic medications that people take, so you can easily help them navigate through that process. Because again, we're here for our customers. That's what we're here for. Skipping on over to page 3, history of medicines and their uses. I'm not going to get into that in a lot too great detail here. If you'll look, the origin of drugs on the right side of page 3, pharmacognosy is a term derived from the Greek word pharmakon, and gnosis is the branch of science dealing with the study of natural origin of drugs. Pharmacognosy is the study of the constituents of natural drugs responsible for their effect. Drugs, definition, is a substance that affects the normal function or structure of humans or animals and can be used to diagnose, treat, mitigate, cure, or prevent disease. They can come from natural or synthetic origins. Of course, natural just comes from nature. Synthetic are going to be the ones that are man-made, that we're making up in a lab. Natural drugs can come from plants, animals, or minerals. Some natural drugs are administered in the crude form. Most of them, the chief active ingredient is extracted from the crude source, and it's kind of made that way. You'll do see the tech notes. So this course has a lot of tech notes as we go through here. And they're all kind of good little tidbits of information for us. Some will be more important than others, and I'll kind of stress that as we go through the course. Wish I could look right into the camera, but the way this thing's set up, I'm not. But, so you see the tech note there on page 3. Some common beverages and foods are natural drugs. Coffee and tea, which we've all heard of those, contain the drug caffeine. Ginger and peppermint contain ingredients that help reduce nausea. So those are just some of your common things that we have. I don't know if you guys know about ginger and peppermint, but if you're feeling nauseous and you get some ginger chews, or you get some peppermint, just plain peppermint, the little round candies, or even a stick of peppermint, it really does help reduce nausea. So if you're car sick, or even if you're just feeling nauseous, those are a couple of good things to use. So what is pharmacology? Bottom of page 3. Pharmacognosy and pharmacology are both sciences that involve the study of medical substances. However, the science of pharmacology involves the action of drugs on humans and animals. The aim of drug therapy is to diagnose, treat, cure, or lessen the symptoms of disease. The study of pharmacology applies knowledge of properties of drugs, mechanism of action, anatomy, and physiology and pathology. If you'll skip back to page 3, there's a chart on the bottom of the page. Couple of call-outs from that chart. It just has some drugs and their sources. So you'll see at the top there, classification as plant. There's a couple of just big ones that we see over and over. Digitalis, the top one, the source is the foxglove plant. Digitalis is the name of the medication that's derived from that plant. In that same little category there, 2 places down, you got the opium poppy, which is morphine is derived from the opium poppy. So the poppy seeds are ground and dissolved and used to make morphine. Further down there, you'll see synthetic. I just highlighted this one as relevant. So under synthetic, you have a synthetic opioid, which is fentanyl. So unfortunately, I'm sure everybody knows about fentanyl. It's pretty popular these days for the wrong reasons. But that is an opioid, and that's a synthetic opioid, and that's fentanyl. All right. So skipping on to page number 4. What is, well, let me go back over here. I'm sorry, on page 4. The selection of the appropriate drug for a patient in the proper dose and dosage form, administered at the appropriate dose and schedule, requires knowledge of pharmacology. The dose is the amount of units given for a single administration. And so the dose is going to be what we're given at one time. They give a couple of examples: 2 tablets, 1 teaspoonful. So a lot of times we're under the impression if I have a tablet, that tablet is 1 dose. The tablet is 1 dose if every time you take it, you're just taking it 1 tablet. If for some reason you're taking 3 tablets twice a day, then every time you go to take a dose, you're actually taking 3 tablets. So your dose for that particular medication in that particular order would be for 3 tablets. So let's say you're doing prednisone, 10 milligrams, 3 tablets twice a day. The 3 tablets you're taking twice a day, so that 3 tablets will be your dose. So your actual dose would be 30 milligrams because each dose is 30 milligrams. So your daily dose would be 20 because you're doing 3 milligrams twice a day. But your actual dose will be the 30 milligrams. The dosing schedule is the number of times the dose is administered per day. So once a day would be your dosing schedule. Once a day, twice a day, 3 times a day. We frequently use the term frequency as well to say how frequently we're going to be taking the medication. Is it twice a day or is it every 4 hours or whatever? So we use dosing schedule and frequency kind of interchangeably. All right, so what is pharmacotherapy? It's defined as the use of drugs in the treatment of disease. Contrasting philosophies about pharmacotherapy exist. You probably need to note the last sentence of that paragraph, which is over on page 5. It says, in homeopathic medicine, drugs are administered in minute quantities to produce effects similar to the disease in a healthy person, yet stimulate the body's natural healing system in individuals with disease. So from that, a loose correlation would be found with immunizations. Because the purpose of immunizations, we give a small dose. This is, again, we're oversimplifying, but we give a small dose of the actual disease to the patient. And we do that, and we give it small enough that they're not going to get sick, but we want to give it a high enough dose that your body will respond to it and develop antibodies, so that if your body sees that later, your body recognizes it and can attack it. And that's the same overall theory with homeopathic medicine. Research and development. So I hate history, but unfortunately it's part of what we have to do here. 1906, Pure Food and Drug Act was passed to protect the public from ineffective and harmful drugs. It was expanded in 1938, and the standards for allowing new drugs were set. The next paragraph there's a lot of steps in the drug development process, and you'll see in Figure 1.1 all the steps that they have. The steps from the test tube production and distribution of a new drug involve preclinical research, clinical studies, the new drug application process, and review. The manufacturers of new drugs have to submit data showing their drug is reasonably safe before a preliminary small-scale clinical study is approved. Preclinical research is conducted to determine a pharmacological profile for the drug and acute toxicity of the drug in at least 2 species of animals. Upon completion of the preclinical phase, drug manufacturers file an IND, which is investigational new drug application, and then you move to the clinical trial phase. Next paragraph, there's 3 clinical study phases, which we're going to cover in just a minute. In phase 1, the drug is administered to a small number of healthy volunteers who enrolled in the study. Preliminary information about the pharmacology, how it works, and how effective it is gathered in phase 1. Excuse me. In phase, if appropriate, it goes to the next phase, which is phase 2. Phase 2 is control trial with a limited number of patients with a condition to be treated. During this phase, data is collected to determine the drug's efficacy and the drug's side effects in patients with the disease. So basically, efficacy just means, is it effective? So we're just looking to test, is it effective in people with the disease? And we're always wanting to look for side effects. And we go to phase 3. If that all goes well, we go to phase 3. Phase 3 clinical studies involve hundreds to a few thousand patients. Drug safety is evaluated, and the benefit of taking the drug is compared to the risk associated with taking the drug. If safety and efficacy are shown, the drug begins the new drug application process and then moves on to review. And you'll see here an accelerated process exists to speed drug development and review when the drug provides significant benefit over existing therapies or a life-threatening illness is present. So we'll continue just to finish that. Additional safety information is collected in the post-marketing phase. Phase 4 trials are then conducted after it's marketed to the public. Basically, drugs are in phase 4 pretty much forever. And that's just to make sure that it's safe and effective once it's used in the real world. So we have phase 1, phase 2, phase 3, and what you'll see is we look for safety in all 3 of those. Phase 1, we just put it in healthy people with that don't even have the disease or condition that we're treating. Phase 2 and 3, we do a smaller group and then a larger group, and that's basically phase 2 is going to be to say, is it safe? Is it safe and effective? When we get to phase 3, the same thing's going to go on. You still look at safety and efficacy, and you also in phase 3 kind of dial up the dosing. So phase 3 is when we'll look and determine is is the dose need to be 40 milligrams or 45 milligrams, 50 milligrams. Because the higher the dose, the more side effects you're going to have, regardless of the medication. The higher dose, the more side effects. So we want to find that balance between, do we have a dose that's high enough to help people, but not high enough to have an extreme amount of side effects. Let's see, we don't need to know that part. All right, over to drug nomenclature on page 6. All drugs are identified by a generic name, a chemical name, and a proprietary name. The name of the new drug is made according to standards set by the Center for Drug Evaluation and Research, or CDER. The standards published in the CDER Data Standards Manual are used by a drug product reference files and the drug registration listing system. Tech note there, every single drug has a generic name. However, that doesn't mean the drug can be manufactured by a generic manufacturer until the patent expires. We'll cover that in a little detail in just a second. So that next paragraph there, the last 2 sentences, the ending of the official name of many drugs indicates the pharmacological class in which it belongs. For example, many local anesthetics have the common indicating. So we'll draw your attention to that table 1-2 on page 7. You'll see a lot of examples, and we're going to go through this in detail as we go through this book in this class. But you'll see that there's just a lot of, what I would ask for you is to let's try to look and study and learn the trends that are there. So benzodiazepines on table 1-2, benzodiazepines are going to end in either Zepam or Xalam. Diazepam and alprazolam are the 2 examples they give there in the table. And there's just there's just tons. Triazolam, alprazolam, clonazepam. There's just a bunch that are there. But my goal would be that we look and we can see that drug, and like we were speaking earlier, if somebody calls and wants a medication and they say, hey, it's my nerve pills or my anxiety pills, you may not know that clonazepam is for anxiety, but my hope would be you could connect the dots, and eventually you will just know clonazepam is for anxiety. But for right now, you can probably connect the dots and say a benzodiazepine ends in Zepam, so does clonazepam, and I think that's going to be your nerve pill. And you can confirm that with the patient and go ahead and move on from there. On page 7, comparison between brand name drugs and generic drugs and biosimilars. So the innovator of a new drug can apply for patent protection. If awarded, the manufacturer is given up to 20 years of exclusive rights to manufacture and distribute the drug. The manufacturer selects a brand name for the drug according to CDER recommendations. When it's off patent, other drug companies can make a generic equivalent. Generic drugs have the same active ingredient in the same strength, same dosage form, can have different inactive ingredients. So, They may not use the same binders to hold the pills together, but they have the same thing, and you can give you the same effect in your body. The next paragraph there, generic drugs are always less expensive than brand name drugs, and that's always the case. So we'll kind of touch base on that really quick. So what happens is when you have a new drug, companies have to spend a lot of money in research and development and getting this drug and going through the clinical trials. And there's tons and tons of drugs that never make it to even seeing an IND, an investigational new drug application, that never make it to a clinical trial. 1, 2, 3. There's a lot of drugs that never even make it to that point. Unfortunately, even though they don't, the manufacturer that has spent a lot of money and time doing research and development and kind of needs to recoup some of that money or they'll go bankrupt. So unfortunately, that cost is also factored in. So when we do have a drug that's actually successfully gone to market, it looks like it's something that's going to work out for us. We do have to, the manufacturer does have to recoup the cost of the research and development for that drug, but they also need to factor into that somewhat of an ability to recoup the cost of the other medications that never made any money for them. So we try to do that and get that going. Generally, you apply for the patent once you start the trials, and as a general rule, you got about 10 years of using your patent before the generic drugs hit the market. So when the generic drugs hit the market, we talked about an INDA and then an NDA, which is a new drug application. But when a generic drug hits the market, it uses an ANDA, which is an abbreviated new drug application. And basically what that does is says, hey, we're going to, trying to think of it, we'll pick Viagra. So Viagra for the longest time had the market cornered. They were the only drug in there for erectile dysfunction. It costs an arm and a leg to get. They kind of charge whatever they want to and still sold a ton of pills. And that was fine. But after their patent ran out, the second it ran out, in fact, they'd already had it planned out before it ran out, and everybody was already stocked up in the warehouse, but they couldn't release it and couldn't sell it. But once that patent ran out, the generic manufacturers could come up and could make generic Viagra, which is sildenafil. And really, all they got to prove is, our pill is the same as their pill. So what they don't have to prove is they don't have to go through clinical trials. They don't have to do an investigational new drug application. They don't have to do any of that stuff. They don't have to go through the research and development. They cut off all that. They cut off all the expense that was done with making the other medications that never even made it to market. They don't have to deal with any of that. All they have to do is say, our pill for sildenafil 100 milligrams has sildenafil 100 milligrams, and your body's going to absorb it at the same rate, and it's going to have the same effect in your body, and we're good to go. So that doesn't take a lot of research and development, time and energy costs into doing that. And because of that, your generic drugs will be bought at a fraction of the price of the brand name, and that's the reason. So drug legislation. So on page 7, drug legislation. So Pure Food and Drug Act 1906 was the first significant legislation passed to protect the public from harmful and ineffective drugs in the U.S. Over the years, they've had a whole lot of other laws passed that regulate manufacturing and distribution. 1951 Durham-Humphrey Amendment established a distinction between legend drugs and drugs that could safely be used by the public without supervision by a healthcare provider. Legend drugs are drugs that can only be obtained by a prescription, whereas over-the-counter drugs do not require a prescription. So basically, in a nutshell, 1951, they said we have prescription drugs and over-the-counter drugs, and they said those are the two big categories. And if it's a medication that you really don't need a medical professional to oversee, you taking it and dosing it and all these things, then we'll make that medication over the counter. And if it does require some oversight, then we're going to make it a prescription or the term legend drug. You'll see in here the Kefauver-Harris Amendment in 1962 requires all drugs to be safe and effective before they're made available to the public. And I know that sounds kind of crazy, but before then, they didn't have that. That wasn't the law. You'll see on down here, I've already talked about it, but a patent. So you'll see a patent. On further down the paragraph, a patent gives exclusive production rights to manufacturers of proprietary drugs for up to 20 years. And that's 20 years from when they apply for the patent. Once it's expired, manufacturers of generic drugs can produce and market that drug. Generic manufacturers are not required to conduct additional studies to prove safety and effectiveness, but instead are permitted to rely on the safety data submitted by the manufacturer of the proprietary drug. So they can just kind of piggyback on the first thing that was sent in. So a little bit easier and cheaper for them to do. Skipping over to page 8. You'll see the first full paragraph there. In the U.S., Combat Methamphetamine Epidemic Act, which is the CMEA, was passed in 2005 to curb the illegal manufacture and use of crystal meth. So, obviously we've had drug issues going on in this country for a long time, and one of them has been and still continues to be crystal meth. One of the things they try to do is to figure out, we're struggling to try to control the production of meth and how are we going to do it. And one of the ingredients that's used to make crystal meth is pseudoephedrine, which is Sudafed. It's the same Sudafed you take for congestion. And back in the day, before this, I can tell you we used to have people, and some pretty sketchy people would come in, and those people would be purchasing a full shopping carts loaded to the brim with Sudafed. They come and drop $400, $500 worth of Sudafed, and pretty obvious what they're doing, but there was no laws to stop it. So kind of put everybody in a tricky situation. If you want to intervene and you want to stop that transaction, you don't really have a lot of good grounds to do that. So they came up with this, and it's, hey, well, if we slow down their access to Sudafed, then we, by definition, will slow down the access and production of crystal meth. And it has worked. I will tell you, any product containing ephedrine or Sudafed, or either one, ephedrine or Sudafed, is kept in the pharmacy. Has to be kept in the pharmacy, is not kept over the counter like up front. It is sold over the counter. It's available without a prescription. We are required to have an identification. Actually, specifically in Virginia, we're required to have the Virginia driver's license. And the way it's overseen in Virginia is we take your license, we enter all that information in the computer, and then we process the sale. And once that's done, it automatically transmits that to the state police. The state police is the agency that oversees the pseudoephedrine and the purchasing and the sales of that. And that's how it goes. Now they do have, and they did set very clearly, daily and monthly limits for pseudoephedrine, and they're reasonable from the standpoint of one person buying Sudafed should not have an issue and ever go with the limits. Once in a while, you may have somebody trying something, or you may have somebody, I think we have a legitimate issue sometimes. Somebody will get it, and I'm getting it from my husband, I'm getting it from my wife and myself, and next thing you know, they're getting too much. But that's because multiple people are taking it. But at the end of the day, it's still the law, and the law is the law, and that's what it is. So, that's the CMEA. The next paragraph, the Comprehensive Drug Abuse Prevention and Control Act, which is the CSA, which is a Controlled Substance Act. It was passed in 1970. It regulates drugs that have a history for abuse in the U.S. Access to controlled substances is more restrictive than access to legend drugs. Controlled substances are placed in C1, 2, 3, 4, and 5 categories to their potential for abuse and effects if abused. Schedules are determined by federal and state law. So there is a table on page 8 that I will draw your attention to, and it does a pretty good job. It kind of breaks down classes 1 through 5. And you'll see class 1, the drug has a high potential for abuse. The drug has no currently accepted medical use for treatment in the United States. And they put the caveat in here under dispensing rules only with approved protocol or investigational use. And that is true, but the only thing that really possibly would apply to being that it's okay to have would be marijuana potentially. The problem with that is, regardless of states and where we are right now, it's still a federal crime, and you are not to be in possession of marijuana. Clearly, PCP, heroin, ecstasy are not going to be or Molly is not going to be things that you need to be having on your person, and those are class 1. There's just no real reason for you to have that. And basically what's going to happen is, as we go down this list, every class we have is going to have a little less potential for abuse and a little less to potential for dependence. So C2, Class 2. C2, Class 2, Schedule 2, it's all the same thing. They use a C and they use a class and a schedule. C2 is if a drug has a high potential for abuse. Abuse can lead to severe psychological or physical dependence. Has an accepted medical use for treatment in the U.S. Dispensing rules, they have to be written or sent electronically. So this thing changes all the time. And right now in Virginia, they can still be written, but they really prefer electronically. And I would assume that in the next year or two, it's going to be all required to be electronic. That just cuts down on the possibility of forgeries. A few years ago, forgeries were the hot button in pharmacy. Anybody would try to call in a prescription and write a prescription, and they did it all the time. So you see some examples of that: methylphenidate, codeine, meperidine, oxy, secobarbital, hydromorphone, amphetamines. You go to C3s, and I guess we'll just go quickly though. 3, 4s, and 5. So 3, potential for abuse, less than a C2. Abuse can lead to moderate or low physical dependence or high psychological dependence. Has accepted medical use. You go to C4 and 5, and it just kind of cascades on down. So we're not going to belabor all that. Any prescriptions for a controlled substance, whether it's a 2, 3, 4, 5, are only good for 6 months, period. If it is a Schedule 2 medication, you're not allowed to have refills on it. I will say if the doctor writes prescriptions and it has refills, we can still fill the prescription. We just have to basically void out the refills. 3, 4s, and 5s are good for 6 months and can have up to 5 refills. Occasionally, somebody will write a 3, 4, 5. They'll write somebody a Xanax and they'll put 7 refills on it. And again, it's legal to fill the prescription. It is legal for us to put the first 5 refills. So even though the prescription may say, number 60 with 7 refills, we take it, we fill it for number 60, but we just cut that 7 refills down to 5, and then we go ahead and process the prescription for it. A C2, you'll see there has no refills allowed because we don't want outstanding pills not knowing where everything is. So they really want to have a very hands-on approach to C2s. We'll talk about this some as we go through, but C2s are generally kept in a safe. C2s are always managed by a per pill perpetual inventory, which means at any given point, I can tell you in my particular environment now, we do it electronically, and it used to be on paper log. Just with technology, most of it's going to electronic record keeping. If the board inspector comes in, which we do get inspected periodically, the board inspector comes in from the pharmacy board and says, hey, and it's something she typically will do just to see how your record keeping is. Hey, tell me how many oxycodone 10-325 you have, and I'll look out on the computer. I'll say 462, and she'd say, okay, let's go to the safe. So we go to the safe, we open it, I take it out, I count it, and I say 462. She said, all right, you're good. And she'll do several of those just to make sure our record keeping is where it needs to be. So that's that. All right, so down to page 8, drug dosage forms and delivery system. So drugs are formulated by delivery by mouth, injection, inhalation, or topical application to skin or mucous membrane. I feel like I'm beating a dead horse with that. So on over to page 9, dosage forms for oral, which is enteral administration. Oral administration is safe, easy, and generally more economical than parenteral administration. Common oral dosage forms are tablets, capsules, solutions, emulsions, syrups, suspensions, and elixirs. A lot of stuff. So basically, tablets are generally, I don't know if it's necessarily safer, but it definitely is easier and cheaper generally than an IV. And IVs are a lot easier because nobody wants to sit around and give themselves a shot if they don't have to. Much easier to swallow a pill. And because of that, when we make a pill compared to an injection, injection has to be sterile. We got to be careful in how we're making it. There's a cost associated with a syringe for the injection. So it does make it more expensive. With a pill, you don't have those things to deal with. It doesn't have to be made in a sterile environment. You don't have to dispense it with a syringe or any specific thing in order for you to take it. So it's just a cheaper way to do it too. So we look under tablets. So there's different kind of tablets here. We have repeat action tablets layered. So basically, you have a tablet that's in layer, and it's going to dissolve, and as it dissolves, it releases certain amounts of the drug as it's dissolving. Delayed action tablets slow the release of the drug to avoid stomach upset, improve absorption, or prevent drug destruction in the stomach. So there's a lot of drugs that once we take them, the stomach acid just degrades the drug really quick. It has a bad degradation. And if you get a delayed action tablet, it'll allow that coating to stay on till the medication, till that tablet is past your stomach, and at that point it dissolves, and then the medication is absorbed. Enteric coated tablets are pretty much the same thing, to be honest. Sustained release or time release tablets, basically what they do is they deliver their contents over time, and there's different ways they do it. I don't know. There's just different ways. And one of the better, I guess, examples of a sustained release or time release tablet would be ProCardia XL. And basically what that particular one is, it's used for blood pressure. And again, but the way they do the sustained release and time release is if you take the pill and look very closely at it, there's a little bitty hole about the size of like a needle, like a sewing needle. About that size that's in the pill. You have to really look to even see it. But that pill, the hole is in there, and basically what happens is that pill, as it goes through your system, that hole is so small, it allows the drug in a time-release manner to come out of the pill and be absorbed into your body. And that's one of the ways they do time release, which I always thought was neat. Film coating and sugar coating tablets is basically designed to help them go down better. So if you have a tablet and it's film coated, it's just smooth, and so when you swallow it, just goes right down. Same thing with sugar coating. On to page 10, chewable tablets are formulated for people who have difficulty swallowing. I think that's a given. Sublingual and buccal tablets dissolve in the mouth. So sublingual tablets are designed to be dissolved under your tongue. And I think that's pretty straightforward. That's what they are. Buccal tablets are designed to be put in your cheek, kind of like you're having some chewing tobacco. You're going to put it right here in the side of your cheek, and it's going to be absorbed through your gums there. You have ODTs, or dispersible tablets, similar to sublingual tablets, but they dissolve faster, and you place it on your tongue. You don't have to put it under your tongue. So sublingual is designed to put under your tongue. ODTs are just designed to be put on top of your tongue. Pastels are solid medicated preparation dissolved slowly in your mouth. Troches and lozenges are dissolved in your mouth. And generally, like the best example is we have a product called MySelects Troche, and basically it's you as far as taking it, you take it like a cough drop. You just take it, you put it in your mouth, and it will dissolve. And basically what it is it's an antibiotic, and it uses a lot for different things. The main thing for it is going to be thrush. So if you have a fungal infection in your mouth, you take that and you let the tablet dissolve in your mouth, and through it contacting the area, it helps treat that thrush that's there. Lastly, we have a thin film, which is a thin layer of film that disintegrates or dissolves to release the drug and apply it to sublingual tablets. So basically, the best example I can give you for the film, and they actually have a product that's called a film, and they can put it in different places. We have some medications that are taken as films. Unless you ever had the Listerine films that you use for your breath there. If you take those and put it in your mouth, that's a film. You put it in your mouth, and as soon as you put it in there, it just dissolves. We have medications that dissolve as a film as well. Next one is capsules. Capsules are pretty straightforward. Solid dosage form containing one or more active ingredients plus binders and fillers. And there's a giant picture of a capsule there on page 10, but I'm sure everybody knows what a capsule looks like. Oral liquids include suspensions, solutions, syrups, elixirs, tinctures, and emulsions. Typically, they're water-based. Drugs formulated in liquids are easier to swallow because the liquid's a lot easier. And they work faster than tablets or capsules, and that's because once you take them, we don't have to wait a little bit for your body to break down the tablet or capsule. The liquid's already there, and it's ready to be absorbed, so it does go quicker. They have some examples there. Suspensions, which has small drug particles suspended in a solvent. The thing with a suspension is, I'm trying to think of an example. If you ever make Kool-Aid, you make it too strong. And every time once you stop shaking it, you'll see all the Kool-Aid settle down at the bottom. And if you're going to drink it, you want to shake it up real good so you can mix it. That's a suspension. So a suspension will it will stay in solution a little bit when you shake it, but once it sits down, it's going to separate out, and your drug particles are going to sit at the bottom. So a big key in the world of pharmacy and a pharmacy technician's role. So a big key for us is to make sure that if we're dispensing a suspension, first of all, that we place a shake well label on it, but most of that's done through our computer system now. Second, and more importantly, is if you're dispensing a suspension, you need to shake it really well before you go to measure it out. Because if you don't, you can have uneven drug particles in your final suspension. So, if I have a bottle of a suspension and say I've got 8 ounces of it, and I'm going to pour 4 ounces of it out, and if I don't shake it, the part that I'm going to color out is not going to have most of the drug. The part that I pour out is going to be mostly just the solvent and just a suspending agent. And then, unfortunately, the person who gets the second half of that is going to get a lot of medicine. And, a lot of the medicines we deal with, and again, we want to treat everything like it's important. We treat every medication, every prescription like it is a life-saving medication, even though most are not necessarily life-saving. Most are helping us improve our quality of life and help extending our life. We have, what comes to mind as an example that I had once had a partner, and we were checking prescriptions together, and there's 2 of us there working, and I looked and I kept noticing, and I'd never seen this before, and she was taking a prescription and kept holding it up to the light, and I asked her, I was like, what are you doing? And she said, well, I have to do this, and I said, I don't understand why you have to. And when she said it, it made perfect sense, and I started doing it too. She said, well, you got to watch people. And she said, unless I see a technician specifically shaking a bottle, she said, I hold it up, and I want to make sure that we got what I believe to be the right amount of drug in there. And I said, oh, that's a great point, And in particular, the one we were talking about, the prescription we were talking about, was actually for a seizure medication for a child. So it was critically important that we did have the right amount of medication in there, and it was important that when we poured it out that it had been shaken so it would be distributed evenly as we did that. So moving on, we have syrups there, which contain a high concentration of sucrose or other sugars, elixirs, which contain alcohol 5 to 40%, and then emulsions there at the bottom, which are similar to suspensions. Suspension generally is going to be a solid in a liquid. An emulsion is going to be like a oil and water. And I give the example, I'm not sure how many of you are familiar with like oil and vinegar dressing. But whenever you use it, you want to shake it up, and it'll stay as an emulsion, which would be like oil and water. It will stay in that emulsion, like in that form, for just a few minutes. And again, if it sits for a little while, you got to shake it up again. Same thing with emulsions. You got to shake them up again. If you're going to pour one out, you got to shake it up again. It's critical if we're giving it to a patient. We got to tell them before they use it. Got to shake it up. Moving on to page number 11, dosage forms for topical administration. So you see there, so topical administration, solutions, suspensions, and emulsions are also formulated for topical application to the skin, the eye, the ear, and the mucous membranes of the rectum and vagina. Topical dosage forms not previously described include ointments, creams, and suppositories. And there's a tech note there that I cannot stress the importance of, and that you really need to know and understand. Eye drops, which are sterile, can be placed in the ear, but ear drops, which are not sterile because they don't have to be sterile to be placed in your ear. So ear drops should never be placed in the eye because they are not sterile. It is a huge safety issue. It will, it could lead to a lot of things. The guarantee is it will lead to an excessive amount of burning and pain. And were you to continue to do it, it could do damage to the eye. Typically, if somebody does that one time, they're fully aware that something is clearly not wrong. And you will have an irate customer on your hand, understandably so, because you could harm somebody doing that, and that's just a huge thing. But also do know it is frequent that we actually do use eye drops in the ear, largely due to cost issues. Ciprofloxacin, we have a Cipro drop that's in the ear, and we have a Cipro that's in the eye, for reasons that make no sense to me at all. The eye one's a lot cheaper than the ear one. So it pretty frequently, if we get one for Cipro eardrops, due to price, we'll call the doctor and follow up and say, hey, can we use the eye ones in the ear? And they say, yeah, we do that and save them some money, because it helps them. It's the same medication, same strength, just saves them some money. So going down here, dusting powders, we don't need to worry about. Ointments are semi-solid preparations containing petrolatum or other oily-based. Petrolatum is just petroleum jelly, Vaseline. Basically, the way I explain ointments and creams, so creams are semi-solid emulsions. The way I explain ointments and creams, if you apply an ointment, pretty much no matter how much you rub it, it's going to leave a wet sheen on your body. If you apply a cream, if you rub it in, it will rub all the way in. That's the big difference. Let's see. Hydrogels contain up to 99% water, control release drug delivery system. They're using transdermal implants and then in wound dressings. And then we have suppositories there. Suppositories are solid or semi-solid dosage forms intended to be inserted into a body's surface. And you'll see the tech note there. I see the tech note there. First, the auxiliary label for topical use only should be applied to topical products that are dispensed. More importantly, and I'll spare you the gross stories which you can imagine, which are true, patients should be advised to remove the full wrapper or outer wrapper from a suppository before inserting it into the rectum or vagina. I've had, on more than one occasion, I've had that question asked of me about why it's hurting. And it's in all seriousness, not even a practical joke. They're very serious and want to know why it's hurting, and that doesn't seem normal. So we do need to make sure and instruct a patient, if that's the case, that they do need to take the wrapper off before inserting. Transdermal drug delivery system, which are patches. Patches are control release devices that deliver medication across the skin membranes into the general circulation. They produce systemic effects, which means they basically are getting affected throughout your body, as opposed to a local effect, which would just be if you put a patch on for pain and it's going to control the pain right there where you're applying it. That's going to be a local effect. What we're talking about is really a transdermal drug delivery system, which is basically allowing that medication to get into your body and to be absorbed into your bloodstream. Dosage forms for parenteral administration, on to page number 12. Parenteral drugs are injected or infused, which is just a slow injection, directly into a blood vessel, muscle, skin, or joint. And you can see there in figure 1A, and just give some examples of some injections. And it gives you the methods that we do, the examples of parenteral drugs. IV solutions are injected directly into a vein, go right into your bloodstream. IM solutions and suspension go deep into the skeletal muscle. Sub-Q go just below the top layer of your skin. Intra-articular solutions are injected directly into a joint. Intradermal solutions go into the dermal layer, which is the very top layer of your skin. And intrathecal solutions go directly into the cerebrospinal fluid in your spinal cord. And again, a tech note, and it's a good one here, to avoid drug contamination. Aseptic technique has to always be followed when preparing drugs for parenteral administration. So that is one of the tricks, or I guess asterisks when you're doing this, is you do absolutely have to follow aseptic technique the entire time, because we cannot have any kind of particles in there or virus or bacteria getting in through that injection site. Dosage forms for respiratory tract administration. Solutions and suspensions are applied to mucous membranes of the nose as a spray or a drop. Have microfine powder solutions and gaseous drugs are inhaled into the lungs using the MDI, which is just a meter dose inhaler. They have it, it says in figure 1.9. Oh yeah, you can see a young lady there in A there with a MDI. It's just a meter dose inhaler. Basically, MDI basically just means that every time you push it, we're going to get the same amount of drug that comes out. So one push gives you the same amount of drug. Even if you push it and hold it for 10 seconds, you're only getting that first same amount of the drug that's in there. And also it says you can use a nebulizer. So a nebulizer is just a device that turns a solution into a vapor that can be inhaled. And they come in different sizes. And basically what happens is we'll give a medication in there, and the medication will be in the form of a liquid. And it turns that liquid into a vapor, and then we breathe it in. So usually used with asthma, COPD, or something along those lines. It works better than an inhaler. It's just less convenient because you got to be sitting down. You got to plug it up. An inhaler can just go in your pocket or in your purse, and you can take it wherever you want. Routes of administration. Formulation of a drug is largely controlled by the chemical properties of the drug and human physiology. The method of drug administration is determined by the drug dosage delivery form. The next paragraph there, the major routes of drug administration are enteral, parenteral, inhalation, and topical. Enteral, parenteral, and inhalation routes typically produce systemic effects. Systemic effects extend beyond the area of the drug application or administration. So drugs applied topically usually produce a local effect, which means it's predominantly at or near the site of application. It's similar, we just talked about the patches. So if you're applying a cream, generally, there's always exceptions, but as a general rule, if you're applying a cream, you're going to apply it to the area it wants to affect. So if you have a rash, you're going to apply that cream to that rash to treat that rash. Or you may apply a cream to help with pain, and you're going to apply it to the area that's hurting. If you're talking about something else, which would be, an IV or an inhalation, that's going to be something that's wanting to get into your system and actually affect the inside of your body, not specifically where it's administered. All right, so back to 13, enteral drug administration. Oral administration is safe and easy compared with parenteral administration. Doesn't need special techniques for administration, and if needed, the drug can be removed from the body by vomiting or binding with activated charcoal. Drugs available for oral administration are generally less expensive. All orally administered drugs have to disintegrate, and dissolve into solution before they can be absorbed. This is a pharmaceutical phase called drug disposition. Tablets and capsules contain binders and fillers that can influence the rate and extent of disintegration and dissolution. The disadvantages are variable absorption and decreased bioavailability. So basically, when we talk about variable absorption, with food versus without food, your GI transit time. So some people's GI transit time might be 2 hours. Some people's might be 20 hours. Because of that, if you have a pill that's going to be absorbed only when it gets to the small intestine, one person might see that result in 20 minutes, as where another person might not see that result for an hour and a half. So that creates a lot of just variation in it. Bioavailability is the fraction of the administered drug dose that enters the systemic circulation and is able to produce a local effect. The extent to which that drug is absorbed and distributed to the site of action can be influenced by the presence of food, which can delay the absorption of some drugs and aid in the absorption of others. The first pass effect, which is really important. So first pass effect is a process where the liver metabolizes a fraction of an administered dose of the drug before it passes into the general circulation. Also reduces bioavailability. And we're going to cover that a little bit in the next chapter. And there's a tech note there. It's important if you have a medication that requires an empty stomach or requires you to take the food that you put the appropriate label on it. So there's other disadvantages to oral administration. Drugs that are swallowed have to pass through the GI tract. Stomach contents contain powerful acids and enzymes that decrease the amount of drug to be absorbed or inactivate a lot of the drugs. Parenteral drug administration is preferred when the patient is unable to swallow or is having nausea and vomiting, or the drug's poorly absorbed by the oral route. Medicines that are administered parenterally bypass the GI tract. So you kind of scoot, typically you got to get your drug, it's got to get absorbed from the GI tract into the bloodstream. If you're going to give it straight into the bloodstream, you bypass that whole process, you just go straight in there. So I don't know where I left off. The bioavailability of parenterally administered drugs is greater than that of orally administered drugs because more of it's available, because none of it's going to be broken down. 100%. If I inject 40 milligrams of furosemide into my vein, I have 40 milligrams of furosemide in my bloodstream at that point in time. If I take a pill of 40 milligrams of Lasix or furosemide, it is going to be absorbed over time. So I'm not going to even have 40 milligrams at any given time in my bloodstream because it will absorb over a period of time. And as it's still being absorbed, it's already being metabolized and being taken out of my bloodstream. So it's a little bit of a difference there. Let's see. Last couple of sentences there at the bottom of that paragraph, I guess. I'm sorry, I'm confusing you guys. You'll see there's some disadvantages to parenteral administration of drugs. Aseptic technique has to be used when you're preparing them to avoid introducing life-threatening contaminants into the bloodstream. And after drugs administered parenterally, it cannot be recalled through vomiting. So, if you were to take something, you take it by mouth and you're allergic to it, you can induce vomiting and get that medication out of your body. Unfortunately, if you've injected that into your bloodstream, it's in your body and you can't really get it back out. So, the next paragraph there, parenteral drugs have to be administered using specialized techniques, which we kind of know that. Proper injection procedures must be followed to avoid harm to the patient. Figure 110. So if you look on the next page, you'll see figure 110 there, and it gives you some just basic things. And you'll see at the top there, the intradermal just goes right under that top layer of skin. The bottom of that figure, you'll see a couple of examples. Intramuscular, you'll use a longer needle, and it goes all the way into that muscle tissue. The subcutaneous uses a shorter needle, and you can go straight in. Generally, it's less painful to go at the 45-degree angle if you're doing a subcutaneous shot. So back to page 13. Here at the bottom, drugs intended for subcutaneous injection pose fewer risks than IV administration. However, aseptic technique has to be followed. The site must be rotated to avoid complications. On to page 14. Inhalation. Inhalation is the most effective way to rapidly deliver drugs locally to cells of the respiratory tract and into the general circulation. Absorption problems encountered with oral administration are avoided. Some of the side effects associated with oral or parenteral administration are minimized. Inhalation is an effective method for delivering meds to treat asthma and other respiratory disorders. Some medicines used to produce general anesthesia are also administered by inhalation. So I think that's pretty straightforward. Transdermal drug administration. Drugs that are formulated for transdermal administration are applied to the skin to produce systemic effects, such as, and they give some examples here, but just understand we do have some drugs that we put on skin. So even though we have a lot of medications and we don't want to we don't want to overstate what's a rule and what's not a rule on this. But, generally, if I'm going to put a patch on my skin, it's going to be to help me absorb something into my bloodstream. And they give an example here of Nicoderm. So, you can have a patch here for Nicoderm, and that Nicoderm is going to be allowing your body to absorb nicotine into your bloodstream. You can also apply a patch of lidocaine, which is not really going to be, it's very low absorption rate into your body, but it actually will produce a local effect here. So kind of a fine line. Topically administered drugs typically produce a local effect, which is what we just said. So typically if we're going to put a cream or something on, it's going to be to treat a rash or a pain that's right in that specific area. That concludes chapter 1, and we're going to skip right over into chapter 2. See if we can't knock this out. It's pretty short. So chapter 2, Principles of Pharmacology. So page 18, pharmacokinetics. The word pharmacokinetics is derived from the Greek words pharmaco and kinesis. We just said that. There's 4 pharmacokinetic phases. They're ADME, absorption, distribution, metabolism, and elimination. Drugs that are administered must be absorbed into the bloodstream and distributed to their site of action before they can produce their effects. The body metabolizes the drug, which is just basically it processing it and breaking it down. So your body always metabolizes the drug, and then it's eliminated. As the drug moves through the body, it undergoes changes that can increase or decrease its absorption, distribution, metabolism, or elimination. These phases control the intensity of the drug's effect and the duration of action. So skipping over, there's a chart, there's a figure 2.2 there. I'm not sure it's going to come back to it, but I do want to show you the onset of action. So on page 19, I'm just going to talk through the figure 2.2 right now. So basically, if you look at that chart, at that little graph or figure, it's a plasma. It's what we call a dosing curve. So basically, you have the concentration on the y-axis, and you got the duration of action, or the time on the x-axis. And you'll see onset of action there. Basically, what you want to think about is, so when you take a pill, and this example is going to be for an oral medication, it's not for an injected medication. If you inject something into your bloodstream, your initial concentration is high and goes down. The reason this one starts down and goes up is because it's an orally administered medication. So it takes a while for that medication. And when we get to drug concentration on the y-axis, when we talk about drug concentration, we're talking about the actual blood concentration. So we're talking about the concentration of that drug in your bloodstream in a sample that's taken. So as we do that, as you take the pill, you're taking a pill, it's being dissolved in your stomach, it's being broken down, and as it's being broken down, it is being absorbed into your bloodstream. So that's as you see the curve at the beginning. As it slowly goes up, that's the drug concentration in your body going up. Once it reaches, in this case, that red dotted line, that's the concentration of the drug that's required to be in your body for you to actually see a therapeutic effect. So I'll give you an example being a blood pressure pill. And let's say this blood pressure pill is supposed to decrease your blood pressure by 5 points. Once it gets to that red dotted line, now on its way up, it may reduce it by 1 point, by 2 points, by 3 points. But once it's at the dotted line, that red dotted line in this example, at that point, is reducing your blood pressure by 5 points. So that's where it's at. You can see the peak effect is going to be the highest concentration that it's in your body. So you'll see the little blue line there, and where it peaks, that's your peak effect. And then it starts to go back down. So as it starts to go back down, you'll see it crosses that red line. And at that point, you lose the therapeutic effect, which means it still may be reducing your blood pressure, but it's only going to reduce your blood pressure by that 5 points or more while it's in that little area where the red and blue meet the first time and the area where the red and blue meet the second time. So only during right in there is where you're going to actually have your therapeutic effect. After that, you're going to see that's the drug as the line goes down. That's the drug being metabolized and then excreted from your body. So back on page 19, back to page 19 in the text. So go down here and you'll see the length of time it takes for a drug action to begin after a dose. It's called the onset of action. The onset of action is not achieved until it reaches the minimum concentration in the body needed to produce drug action. So I will tell you this chart is wrong. So the onset of action is actually, for this particular chart, the onset of action is right where the red line and the blue line meet. That's the onset of action. Yeah, and this whole picture is wrong. I didn't even realize it. So that's the onset of action. And if we're reading the text down here, the duration of action is the time between the onset of action and discontinuation of drug action. So the duration of action, if you look on the chart, it's just wrong. So the onset of action is going to be the first place where the red line and the blue line meet, and the end of the action is going to be where the red line and the blue line meet the second time. The duration of action is actually going to be this little part right here, and I'm going to see if I can hold this up. I don't know if you guys can see that or not, if it's backwards. So right here is going to be the onset of action, is right here. Right here at the top is going to be the peak effect. And then over here at the other side, so the area here to here is going to be the duration of action. This is not right. This to this to this. That's your duration of action for this particular medication. Can't believe they have it that wrong in the book, but it is. All right, so moving on, pharmacokinetic phases. So the first phase is going to be absorption. Absorption is the first pharmacokinetic phase, the process that involves the movement of drug molecules from the site of administration across cell membranes and into the circulatory system of the body, which is basically going to be, it says blood or lymphatic system, but for all intents and purposes, it's just moving it into your bloodstream. You go on down to cell membrane. Drug movement through the site of administration into the circulatory system depends on the ability of the drug to move across cell membranes. Cell membrane is a complex structure of lipids, protein, and water-filled channels. And there's a figure 2-3 on, I think, the next page. Yeah, and it's kind of busy, but you'll see a figure on page 20. It's busier than I like to see, but that is a picture of a cell membrane or a plasma membrane. let's see, movement across the membrane. All right, so movement across the membrane is restricted unless the drug can pass through the lipid layers of the cell membrane or is small enough to pass through the small water-filled channels. A lipid-soluble drug can easily move across the cell membrane. Drug transport mechanisms. So we talk about passive and active transport. Drug absorption across cell membrane can occur via passive or active transport. Drugs absorbed by passive diffusion move from a region of greater concentration to a region of lesser concentration. When the drug first enters the body, the concentration at the administration site is greater than the bloodstream. Lipid-soluble drugs readily regularly diffuse across the cell membrane of the blood vessel into the bloodstream. Lipid-soluble drugs are called lipophilic or hydrophobic or water-hating. Water-soluble drugs are called hydrophilic or lipophobic, but it doesn't say that. The last sentence in that little paragraph there, most drugs are transported by passive transport. So most drugs use active transport, and the big thing is active transport doesn't require any energy. The way I try to explain this, and I'm not sure if it's going to help you guys on this web thing, if I'll be able to explain it, but I'm going to try. So the way I try to explain the passive transport process is, let's say you have a big aquarium, and you have a divider right in the middle that divides the left side from the right side. And so you've got your aquarium, and that divider has holes in it, and let's say those holes are the size of a small orange, just to have something to say. The size of a small orange. So you have an aquarium, is divided in half. The divider, though, has holes in it the size of a small orange. You fill that aquarium with golf balls. Now the golf balls, and if you just shake it a little bit, the golf balls, though, are not going to need a whole lot of coercion. If you dump them into one side, they're automatically just going to flow into the other side because they're just going to go through that hole because they're a lot smaller than that hole. So they'll automatically just cross that membrane and even up, and you'll have roughly the same on each side. That's passive transport. That's the golf balls. If I pour them in one side, half of them are going to go over to the other side because that's the area of a less concentration. That's passive transport. Active transport is going to be where some kind of energy is required. The example I give, let's say that small orange, so we'll take that exact same example, and let's say that small orange was a little bit smaller than a tennis ball, okay? So we poured the golf balls in there, they went right through. Now if I have tennis balls, I dump a bunch of tennis balls into one side. The tennis balls, again, are a little bit smaller than that orange was, the size of the holes. So in order for me, so when I do that, none of the tennis balls are going to go through the holes because they're too big. So if I want to get those tennis balls, I'm going to have to squeeze them through. And by me squeezing them through would be me applying energy. So, passive transport would mean no energy is required. The stuff's going to be evenly distributed on both sides. Active transport is going to mean that some amount of energy, and in my example, is just going to be physically pushing those tennis balls through that hole because the hole's a little bit too small. That requires energy. So that's the best example I can give you to try, hopefully, to explain that concept. So active transport mechanism permits the drug to move across cell membranes without regard to concentration. Drug can move from an area where drug concentration is low to where it is high. Active transport takes energy and requires special carrier proteins or pumps to carry the drug across the cell membrane. So factors influencing absorption. So again, we're talking about absorption. Effect of pH, and most drugs are either weak acids or weak base. All right, in a solution, a weak acid and weak base exist between ionized and non-ionized state. As far as I'm concerned, we're not going to focus on that. So we're going to move on down to diffusion, the next or the couple paragraphs down. So diffusion across cell membranes is greatest, and that's just when your drug's crossing cell membranes, is greatest when the drug is lipid soluble and non-ionized. When a weakly acidic drug such as phenobarb is in the stomach, it's less ionized and can readily cross the cell membranes. Absorption is high. When it moves in the small intestine, ionization increases and absorption is reduced. Osmosis is the movement of water across a semi-permeable membrane from an area to higher to lower concentration. Effect of blood flow on absorption. Absorption is greatest in the area of the body that has a good blood supply. I think it's kind of just a given, but medications administered sublingual have good absorption because there's a lot of blood vessels under the tongue. Absorption of orally administered drugs is greatest in the small intestine. That's because the structure of the intestine is designed to perform the specialized job of absorption. And typically it's designed to absorb vitamins and minerals and nutrients from food you're eating. But it has thousands of microvilli that line the walls of the small intestine. They're filled with blood vessels, and as the drug passes through there, it gets absorbed. Affect the surface area on drug absorption. It kind of goes to the same thing. Microvilli also increase the surface area of the small intestine, making it the largest absorbing surface in the whole body. And there's a picture there, although it's kind of weird. But affect the contact time at the absorption surface. And that's kind of relatively a given. If you have a drug that's absorbed in the small intestine or large intestine, and you have a rough bout of diarrhea right now, you're probably not going to get that drug absorbed as much as we want because it's just going to kind of pass on through. Effect of tissue thickness on absorption. And it kind of is a given. If you're going through, if you're only having to go through 1/4 of an inch of tissue, it's going to go faster than if it has to go through 1/2 inch. So that's absorption. The D, and we're talking about ADME, A-D-M-E, so we're to the D now. Distribution is the process of movement from a drug from the circulatory system across barrier membranes to the site of action. So this is when it's actually going to work, and it's going to move from the bloodstream to where it's going to have its action. So you'll see down there body fluid compartments and electrolytes. Extracellular fluid contains mainly the plasma found in the blood vessels and the interstitial fluid that surrounds the cells. Intercellular fluid refers to the water inside the cells. The major electrolytes are sodium, potassium, and chloride. They're major ones found in the body. Sodium is the major positive ion in extracellular fluid. You'll see the next paragraph there. Potassium is the other major cation found inside of cells. And the next little paragraph, chloride is the major anion, which is the negatively charged ion found in the fluid outside of the cells in the blood. Factors influencing distribution. So affect of drug properties on distribution. Distribution of a drug across the blood vessel cell membrane, transport to the site of action, is influenced by the chemical nature of the drug, such as lipid solubility. Which I kind of discussed that a little bit. Effect of protein binding on distribution. So the blood contains albumin, which is a plasma protein. Many drugs have an affinity for albumin and bind reversibly to the protein. When the drug is bound, it's unable to move out of the blood vessels to get to the site of action. Colloids, proteins, or other large molecules that are too large to cross cell membranes remain suspended in the blood for a long period of time. And the last sentence of that paragraph, competitive protein binding represents a mechanism for drug interactions. So the way I try to explain this is, so your bloodstream, your blood naturally has proteins in it. And albumin is the name of the main protein that's there. So you got proteins that are in your blood that are just circulating all the time. Your body's just got proteins going around all the time. A lot of drugs are highly protein bound. And what that means is the drug's going to go into your stomach or your GI tract, and it's going to get absorbed into your bloodstream, and then it needs to jump off at the site of action. So let's say it's regulating your heartbeat. Even though your bloodstream's going right there, the drug has to get into your heart muscle. So it's got to jump out of your bloodstream into your heart muscle. And the problem is if a drug is highly protein bound, what that means is once it's in your bloodstream, the tendency is going to be for it to stay stuck to that protein. So it won't let go of the protein. Think of it as a magnet. You got two magnets together. It's just going to keep circling around till eventually it'll probably kick it off. But it will pretty much is going to keep it from getting to that side of action if it's highly protein bound. The other issue becomes, and there's a chart here, or picture, figure 2.6 on page 22 that goes through protein binding, and I guess it's okay, but I'll try to explain it and articulate it. And if you understand that picture, then you're good to go. If you have two drugs, if I have one drug, say I have two drugs that are highly protein bound, and I have two of them, and they're both really bad at, or good, or whatever. They're really strong at binding to that protein in the blood. So, for the sake again of me oversimplifying to try to give you an explanation of this, let's just say there's 10 molecules of albumin in your blood, and let's say I give a drug that has a really strong, it's really highly protein bound, and it's going to get bound up to, let's say 9 of those 10 albumins are going to have the drug attached to it. And so the next drug you give, so let's say you give that drug and then an hour later you give a second medication. That second medication, let's say it's highly protein bound as well. Well, the catch is we assume that drug is highly protein bound. So we give a lot of extra. So that's one of the reasons that, this dose may be a little bit higher, because we go a little higher because the assumption is a lot of it's just going to be stuck in the bloodstream going around. So what we want to do then at that point is you're going to give that medication, but it's going to be, your dosage is going to be different, and it's going to be a drug interaction because the other drug is already stuck to the albumin. So because the other medication is stuck to the albumin, the new medication won't be stuck to the albumin, and it'll be free to roam free in the bloodstream, and now you're going to have too high of a dose of that medication. So I hope that makes sense, and if it doesn't, you can reach out to me, and I'll try to explain it in a different way. I'd be glad to. Anatomic barriers to distribution. We're still on page 22. Natural body barriers can limit the access to the site of drug action. Anatomic structures that selectively limit drug access are the blood-brain barrier, the blood-placenta barrier, and blood-testicular barrier. I'm not going to get into all of these things. What I will tell you is you have those three things: the blood-brain barrier, blood-placenta, and blood-testicular barrier. Blood-brain barrier is probably one of the bigger ones just to understand. And basically what they're saying is your body naturally does a lot of wonderful things, and one of the wonderful things it does is it has a barrier that is designed to keep harmful chemicals from getting into your brain. So if you have a harmful chemical that's in your bloodstream, your body will naturally try to keep that from your brain, and that's the purpose of that blood-brain barrier. The problem is if we're given a medication that we need to get into the brain, we need to design that medication in order to cross that blood-brain barrier and get in there and be able to work. So we have that to overcome. So that's the A, the D, and we're up to the M, metabolism. Few drugs that are administered are eliminated unchanged. There are a couple, but not many. Most drugs are transformed by enzymes to enzyme to a metabolite. Biotransformation is the process of drug metabolism in the body that transforms the drug into a more active, equally active, or inactive metabolite. The primary site of biotransformation is the liver. However, metabolism can occur in the intestines, lungs, kidneys, or other cells. Microsomal enzymes in the liver are responsible for transforming lipophilic drugs to compounds that can be more easily eliminated in the body. And use the cytochrome P450 enzyme or system is frequently used in this process, and we'll talk about that a while. But the catch is a lot of drugs are broken down by the CYP450 system. And the problem is when we give multiple drugs that are all broken down by that same system, it kind of blocks it up, and your drugs will end up being higher in your bloodstream because your body's not able to break them down as fast because there's something else using that CYP450 system. So these drugs kind of more or less are waiting in line to be metabolized, so it can cause higher doses of drugs. Couple paragraphs there, you see latanoprost, which is a really interesting drug. It's for glaucoma. Latanoprost is a drug used to treat glaucoma. It's an example of a drug that's metabolized in the eye rather than the liver. It's also a good example of a prodrug. Prodrug is a drug that's administered in an inactive form and has to be metabolized to its active form. Drugs can be formulated as prodrugs to avoid side effects or to increase distribution to the site of action. So a lot of times, if you give that drug in a prodrug form, it's allowed, or it makes it able to get to where it needs to work and then actually be metabolized to the drug that's going to work. Let's see. Levodopa is a prodrug metabolized to dopamine in the brain. And the reason I'll just read this because we're going to talk about it. The drug is used to treat Parkinson's. Levodopa is able to cross the blood-brain barrier better than dopamine. So, yes, could we give dopamine? Yes. But when we give it in the form of levodopa, it gets into the brain, it crosses that blood-brain barrier, and then it's metabolized to dopamine. So, administration of the prodrug increases the volume of the drug that's actually distributed into the brain. And the drug carbidopa to levodopa further increases the amount of drug that's converted to dopamine because carbidopa interrupts levodopa metabolism outside the brain. So basically, we could give levodopa, and that would be fine. When we give levodopa in a combination with carbidopa, the carbidopa, what it does is it actually stops your body from metabolizing levodopa. It doesn't stop it, but it reduces your body's ability to metabolize levodopa until it's inside your brain. So it allows more of the drug to get inside your brain. First pass effect. Orally administered drugs have to pass into the hepatoportal circulation, which is your liver, before entering the general circulation. First pass effect, and this is a key thing you need to know. First pass effect is a process whereby only a fraction of an orally administered drug reaches the systemic circulation. Much of the drug is metabolized in the liver to an inactive metabolite before it even really gets into the general circulation. So that is the first pass effect, which you absolutely need to know. Next, we have 7 factors influencing metabolism. Effect of liver function on metabolism. Since the liver is the primary site for metabolism, you obviously have problems if your liver is not functioning properly. Effect of disease on metabolism. So, again, if you have hepatitis, which is a disease of your liver, that's going to be an issue. And you can also, because we talked about other drugs are metabolized in the lungs and the kidneys. So if you were to have lung disease or kidney disease, that would be an issue as well. Effect of age on metabolism. Your liver doesn't work as well. The older you get, the less it works. So especially in elderly, you have to do some dosage changes in the elderly due to the fact that their liver is not going to be working at the same efficiency. A 70-year-old and a 30-year-old, their livers aren't going to be working the same way. The same thing with an infant. So if you're an infant, your liver hasn't had time to grow and develop. So if you're an infant, you're going to have a slower metabolism as well because your liver is not going to be up to snuff as far as being able to metabolize drugs in an effective manner. Number 4, effect of concurrent administration of drugs, which is basically drug interactions. So if you have drugs that are highly protein bound, or some drugs you give and they actually will adhere to each other and make them unable to work. Effect of genetics on metabolism, and that's just basically the biggest thing in there is enzymes. Due to genetics, some people just genetically have more enzymes or less enzymes, and the higher or lower number of enzymes affects your body's ability to metabolize things, and it can affect your metabolism. Effect of nutrition on metabolism. So your diet, kind of goes without saying, but your diet will greatly affect your metabolism. And effect of gender on metabolism. So depending on the medication, some men metabolize them faster than women, and some women will metabolize them faster than men, depending on the specific medication. Elimination. So we're at the E for ADME. Elimination is the final pharmacokinetic phase. Elimination results in removal of drug from the body and discontinuation of drug action. 3 major routes are the kidneys, the lungs, and the bowels. And really the bulk of it is the kidneys. The normal function of the kidney is to filter blood and remove things that are foreign and harmful. All right. So 4 factors influencing elimination. The first one is the effect of kidney function on elimination. Kind of goes without saying. Your kidneys are our main drug. If they're not working as well, we're not going to be able to eliminate the medications in a timely manner. Next is effect of drug ionization on elimination. So long story short, changes in acidity in the urine can influence the rate in which a drug is cleared from the body. Weakly acidic drugs are less ionized in acidic urine. As the pH becomes more basic, the ionization of the acidic drugs increases. And it just affects whether they're eliminated quickly or not. Effects of disease on elimination, kind of the same thing. Lung, kidney, and bowel disease, because they all play a role in elimination. So any kind of disease you have in the lung, bowels, or kidneys can affect it. Effect of concurrent administration of drugs. And that's just if you're taking other drugs, they can decrease or increase the elimination. Elimination half-life. So this is important. Elimination half-life. We're on page 25. Refers to the time it takes for 50% of a drug to be cleared from the bloodstream. It takes about 8 half-lives to entirely eliminate a drug from the body. So I get that question a lot. How long does it take this drug to get out of my system? And the answer is it takes 8 half-lives. And if you were to read the package insert for a medication, it would tell you what the half-life of a medication is. And it might be 5 hours, 10 hours, 20 hours. But basically, you take that number, multiply it by 8, and that's how long it's going to take it to get totally out of your body. And the reason you would really want to know that, that I've seen repeatedly, is if someone's allergic to it. You want to know how long it's going to take you to get out of that and to not have that allergic reaction. Bioavailability and bioequivalence of drugs. Bioavailability is defined the rate and extent to which the active ingredient of an administered drug enters the systemic circulation, reaches the site of action, and is available to produce an effect. That's what bioavailability is, and you don't need to know bioequivalent. Basically, bioequivalence just means that two drugs have the same bioavailability. That makes them bioequivalents. Pharmaceutical equivalents and pharmaceutical alternatives. Pharmaceutical equivalents differ from bioequivalents. Bioequivalent drugs and pharmaceutical drugs both contain the same active ingredient and the same strength as the innovator's drug. Pharmaceutical equivalents can have different inactive ingredients, be manufactured in a different dosage form, and have different rates of absorption than the brand name. Pharmaceutical alternatives contain the same active ingredient as the brand name, however, strength and dosage form may be different. The most important thing on this little blip is going to be bioequivalent drugs and then therapeutic alternatives. So therapeutic alternatives may contain different active ingredients yet produce the same desired therapeutic outcome, and that is something that we use on a pretty regular basis in the pharmacy. And that is something that the pharmacy technician would play some amount of a role in helping us work through as well, because basically what would happen is, we fill prescriptions for our patients, and we need our patients to take their medications in order to get the desired effects. And a lot of times, if the patient is getting a prescription for, let's say, Simvastatin, and they're getting 90 pills, and it's $140, and we can convert that over to, let's say, Atorvastatin, and instead of that price, we can bring it down to $24. Is it the same drug? No. Are they both HMG-CoA reductase inhibitors? Yes, they're both statins. They both have similar side effects, similar dosing profiles, similar effects on your cholesterol. They're not the same, but they have similar effects. And the truth of the matter is, if cost is an issue for this person, if they're having to pay $140 versus $24 for a 90-day supply, depending on, every person is different. But if price is an issue for a person, I wouldn't say we just throw it out there. But if someone comes and says, hey, Simvastatin's expensive, anything y'all can do, then one of the big options is, well, we'll try to see about getting a therapeutic alternate for you. And we can contact, now that's not something we can just do. That is something that we would want to call a physician, get with a doctor, explain the situation to the doctor. And I will tell you, if you explain it to the doctor, the feedback's been pretty positive. Because the doctor is a person too, and they have bills too, and they understand it's more important it'd be much better for this person to have Atorvastatin and take it every day than them to have Simvastatin and only take it once a week because they can't afford it. So that's a conversation we have pretty regularly. All right, well, I appreciate it. That's chapters 1 and 2, and everybody have a good one. We'll be talking to you soon. Thank you.